Welcome to Study G. Today we will be doing microbes and industrial products. The organism which we use is again called as Saccharomyces cerevisiae. In this case it is called as brewer's yeast, or you can also call it baker's yeast. Both are one and the same. So we use fermenters, which are large vessels used to ferment alcoholic beverages on an industrial scale. Large tank, so how are the alcoholic beverages produced? So the yeast, what it does is it ferments. The yeast ferments fruit juices and cereals to produce ethanol. The substrate which is used in alcohol industry is called as molasses. So what actually happens? The yeast feeds upon the cereals or the fruit juices, and through anaerobic respiration it forms alcohol. So through anaerobic respiration it forms alcohol. Now here we see, depending upon the type of raw materials used for fermentation and the type of processing, we get various types of alcohol. For example, with the raw material, the beer is prepared from barley malt, while the wine is prepared from fruit juices. Whiskey is prepared from fermented cereals. Brandy is again fruit juices are used. Rum, we use molasses to prepare rum. And gin, we use rye malt. And for vodka we use potatoes. And another interesting thing we should note is the type of processing. So the type of processing also yields different type of alcoholic drink. For example, here the beer and the wine, these two are non-distilled, hence they have very lower concentration of alcohol, while here when we compare it to whiskey, brandy, rum, gin and vodka, the alcohol percentage is very high. This is because they are distilled. Let's see the types of processing. So without distillation we get lower alcohol concentration. Examples can be beer and wine, and with distillation we get higher alcohol concentration. Example, we have brandy, whiskey and rum, etc. So these are the main examples which we need to remember. Coming to the next part, we have about antibiotics. So the chemicals having the ability to kill or retard the growth of a disease causing microbe, these chemicals are known as antibiotics. So the first antibiotic which was discovered was penicillin, and the organism from which we got penicillin was Penicillium notatum. Penicillium notatum, it is a fungus. Alexander Fleming was working with a bacteria called as Staphylococci bacteria. So what happened is, in one of the unwashed culture plate there was a mold which was growing, that is the fungus which was growing. So wherever the fungus was growing, the bacteria could not grow. So he observed that there was some sort of a chemical compound which was released by the fungus that did not allow the growth of the bacteria. So then he named that compound as antibiotics. But however, what happened that he could not completely establish a fully effective antibiotic, and it was done by other two scientists. Their name was Ernest Chain and Howard Florey. They showed full potential of an antibiotic. Unfortunately, Alexander Fleming died before they could show the full potential of antibiotics, and later these two scientists, that is Ernest Chain and Howard Florey, they together showed the full potential of antibiotics. Hence, all of these three scientists were awarded Nobel Prize in 1945. Fleming, Ernest Chain and Florey. So these three of them together got a Nobel Prize in the year 1945. So why did they use these antibiotics? So antibiotics were used to treat soldiers, were used to treat American soldiers during World War II. Antibiotics are used to treat plague, whooping cough are also called as Kali Khansi, diphtheria, that is Galghotu, and leprosy, the Kustrog. Now coming to chemicals, enzymes and other bioactive molecules. So here we have organic acids. So the organism which we use to produce citric acid is Aspergillus niger. Acetic acid is prepared from the bacterium called Acetobacter aceti. Butyric acid is prepared from the bacterium called Clostridium butylicum. And lactic acid, as we all know, lactobacillus is used to produce lactic acid. So in this case, this is a fungi and the rest three are bacteria. Now for alcohol we use again Saccharomyces cerevisiae is a yeast in the commercial production of alcohol or ethanol. Commercial production ethanol. Now coming to lipases, the organism which we use to prepare lipase is called as Candida lipolytica. And the use of lipase, we usually use it in the detergents which remove oily stains from clothes. So lipases are those substances which we use to remove oily stains from the laundry. So this is all about lipase. Now coming to streptokinase. Streptokinase is extracted from a bacterium called Streptococcus. So the main use of streptokinase is as a clot buster. So this clot buster removes blood clots from patients who have undergone myocardial infarction, that is heart attack. So to remove that particular blood clot from the blood vessels, they provide streptokinase to the patients. Next we have about cyclosporin A. Again we use a fungi in this case, that is we use Trichoderma polysporum. Is a fungi. Now the cyclosporin A is used as an immunosuppressive drug. So immunosuppressive drugs are those drugs which are provided to the patients who have undergone organ implants. So to prevent the immune system from attacking those grafts. Now coming to statins. Use Monascus purpuris. Is a kind of yeast. Monascus purpuris is an yeast. So again, it is used as, so statins are used as commercial blood cholesterol lowering agent. So what the statins does is, it acts on the enzyme responsible for the synthesis of cholesterol. And prevents the formation of cholesterol. Next we have about pectinases and proteases. So the organisms which produce these are Aspergillus niger. Aspergillus niger is responsible for producing pectinases, while Bacillus species are responsible for producing proteases. So the use of pectinases and proteases is to clarify the fruit juices. Now if you compare the homemade fruit juices to the ready-made bottled fruit juices, we find that the fruit juices which we prepare at home are usually not that clearer. It has a lot of fiber in them. When you compare it to the ready-made juice, it is a lot more clearer. So usually in these industries, they use pectinases and proteases to clear out the juice and make them a lot more clearer.